Can everybody hear me okay? Alrighty. Well, welcome everybody to the University of Wisconsin-Madison's 14th Annual Plant Sciences Symposium. My name is Audrey Morrison, and I'm the current president of this year's Council for the Plant Sciences Graduate Student Council, or PSGSC. I'd like to start today's event by welcoming our invited speakers, student presenters, industry personnel, and fellow researchers. Every year, we look forward to this event, and your continued enthusiasm and participation make this all possible. Today's symposium information is available on our website, and the program can be accessed via the QR codes on your tables. We have a full day of great talks ahead of us. Be sure to stop by the welcome table for your symposium items such as name tags and merch. Following a few presentations in our lightning talk competition, we'll break for lunch from about 12:20 to 1:30, where lunch will be buffet style right outside those doors. During this time, we will encourage attendees to socialize and check out poster presentations going on from students. We also encourage attendees to scan the QR codes that are on the corner of the poster boards and vote for whichever poster you think was presented well, looks well, etc. We're having a competition for that as well. We even have a badger visitor during lunch and poster presentation hour, so be sure to say hi or snap a photo with Bucky. And after lunch, we'll have a few more speaker presentations, a speaker panel, and we'll announce the winners of the poster presentations and the lightning talk competitions. This year, our council wanted to implement travel awards for students from outside universities to come to Madison and be able to present their research by way of lightning talk. So we want to recognize Ashley Barstow from North Dakota State University and Julian Cooper from the University of Minnesota. Be sure to give them a warm Badger welcome, and thanks to you both for attending this year. There are a couple scheduled breaks throughout the day, but please feel free to take the time that you need and you're able to come and go as you would like. So to help facilitate conversations, we have a name badge ribbon system. Every attendee has a ribbon or ribbons on their name tag with colors corresponding to occupations such as student, government, academia, industry, as well as those participating in today's symposium such as presenter or speaker. If you ever require assistance, you can easily identify a PSGSC board member by the black badges on our name tags or our green polos. So following each speaker presentation, we'll have approximately 10 minutes for Q&A. If you'd like to ask a question, you can just simply raise your hand, and a PSGSC board members on the sides of the room will come in and deliver a mic to you, and you can ask directly into the microphone. But as well, if you would like to type in your question, we have this QR code system, which will be on the screen, but it's also on your table. You can scan in and type your question, and then I will be moderating the Slido website, and I'll look for your questions to come in, and we can also answer that via microphone as well. And any questions that we are unable to answer in that allotted 10-minute time, we can save for the speaker panel at the end of the day, so your question will not be forgotten. So today's symposium is organized by the Plant Sciences Graduate Student Council. PSGSC is an entirely graduate student-run organization made up of representatives elected by our peers from numerous plant science training programs across campus. Throughout the year, PSGSC organizes social and professional development events for graduate students in plant sciences. The Symposia series gives graduate students a fantastic opportunity to organize, host, and create connections with others in the plant sciences community. In 2011, UW-Madison joined the Corteva Symposia series as the second university to take part. PSGSC was the student-led group that took on the responsibility, and now tradition, of organizing and hosting this great event for campus and community members. Planning these events takes several months. Our council started our very first process decision back in February in order to make sure that we could deliver an event to this caliber. PSGSC's Plant Sciences Symposia are unique in which each one reflects the identities and values of the members who organize it, so you won't get the same one twice. We are extremely grateful to be able to host a legacy event, such as the 14th Annual Plant Sciences Symposium. We recognize the honor that it is to be a part of Corteva's University Symposia series, and we're thankful for their support and relationship. This slide also shows all the previous flyers since we started, which is really neat. So you can see how it's evolved. You can see the different councils that planned it, what topics were important at that time for them, and you can see where we are now. So I'd like to turn the mic over to Dr. Jason Rauscher, who is here representing Corteva Agriscience, and we'll talk a little bit more about the Greater Symposia series.
All right, thank you so much, Audrey. It's a pleasure to be here with you. And I guess, do I advance it? Oh, there we go. Thank you so much. It's great to be here in Madison and engaging with Audrey and all of the great student organizers who are putting together this event. I wasn't here last year. I was up on the screen last year, so it's great to be back in person and engaging with everyone here. I just want to take a few minutes both to welcome you on behalf of Corteva Agriscience. We're really excited to again have Wisconsin as part of our program for so many years. But I also just want to talk a little bit. I know some of you are very familiar with the program. Others may not be as familiar. So who are we? This is a global program, right? It is each of these events are independent, student-run, and it's an opportunity for industry, right, specifically us, to support graduate education in kind of a unique way. And it was started back in 2008 by, many of you know, Tabare Abadi, who is still at Corteva. But this is, we have kind of a mixed model of some institutions that do it on an annual basis, others that are kind of ad hoc events, but we've really had an amazing global impact at, I think, 22 countries is still our count, all over the world. And so the question is, why? Why do we do this? And we've kind of boil it down into four aspects. So the first being the learning, and that's kind of the fun part. I mean, Audrey showed all the flyers from the different years. And I love all the creative ways that, I don't know, somehow Wisconsin is always very good at coming up with creative titles, leaves dropping and kind of cool stuff. But it's a unique opportunity for them to curate their own scientific event, invite speakers that they're interested in. And it, as Audrey said, it always reflects the interests of each unique student organizing group or committee that gets together each year. So each year is very different, and that's fun for me. It's about networking. It's an opportunity to be in the room together, to meet people, and we all know that our careers depend on networking, meeting people that you might, line up postdocs with in the future, or, get that opportunity to get an interview, in whatever aspect of it. And for us, exploring the different avenues of career opportunities is so important, right? It's not just about academia. It's not just about industry or just government. It's, all of these together. They're all potential career paths, and really having an opportunity to engage with people from all of these different aspects of the scientific community is critical. And I think this is a unique opportunity to do that. Obviously, we bring a Corteva speaker as part of this program, but we also open it up. I'm really happy that we have lots of other industry representatives here today. So it's really a great opportunity for all of us to talk and work with these students. And finally, this is really what you see on display today: a group of students that have been working for months on this event, who have really been showing, teamwork, organization skills, managing a budget, communicating, problem solving, all of these things that are very important, in addition to the technical skills that they're learning in graduate school. These are the things that we're looking for when we're hiring. So having an opportunity for them to develop these skills, to show it off on a CV, we think that's probably the most important part of this program. And it's a great opportunity for me to meet the students and, hear about their ideas as they're developing them. So again, this, it's a big thank you to all the hard work that they put into organizing this event. And also I just wanted to highlight, you can see evidence of this, even as we first arrived at the hotel yesterday, we had a little bag with the schedules in there. I don't know, these guys are really organized because, our little agenda schedule there had told us, the make and model of the car that was going to pick us up and exactly what time. It's pretty impressive. And probably the best is that I'm a student today. So I am going to carry this with pride that I am an honorary student. So thank you for that. I did want to highlight a couple of things that we've done. These are some of the recent ones. This has been a busy week. We have an event in Argentina going on simultaneously right now. So I chose Madison over Argentina. So there we go. And we had one at Viçosa a little bit earlier this week. And we also had a virtual symposium. And we're kind of at a stage where, we had our in-person, and then we went totally virtual during the pandemic. And then we kind of came back to in-person, and people don't really want to be online anymore. So we're in a weird dynamic of, how do these things fit? And we put together a virtual symposium focused on Brazil in biologicals, which is a strong interest for us at Corteva. And so we hosted this week, and we had over 300 attendees for an event entirely in Portuguese, which was really, really amazing. So there really is an appetite out there for these kinds of events, both in person and virtually, I think. So I think that's very exciting. We also do some other things like our New Frontiers Conference series that I manage. We had one on climate resilient agriculture this past year. We had some very exciting speakers. We had the Undersecretary of Agriculture join us, and a lot of exciting researchers coming together to talk about climate change and agriculture. Our internship program, I want to highlight that. Lots of opportunities for both graduate students and undergraduates. Internship season is, you have to look for those opportunities starting around September, October. That's the time to start looking for them for the following summer. And we have 3-month and 6-month opportunities, and a lot of graduate students. It's kind of growing in terms of the numbers of graduate students that are coming in to do these internships. So I wanted to highlight that as well. And so finally, just to wrap it up, did I cover my time here? Am I doing all right? Okay, good. Reach out to me, connect to me on LinkedIn, send me an email if you need anything. Again, it's a pleasure for me to be here. I have the easy job. I just come up and welcome everyone and give the quick 10-minute thing here, and then I get to enjoy all the great talks and engage with everyone. So have a great day, enjoy, and thanks again to all the organizers.
Well, thanks, Dr. Rauscher. And I did forget to mention that if you do choose to ask your question via the Slido QR, please put the speaker's name and then a colon and then your question, and that way we can make sure we're directing your question specifically to who you like to ask it to. So our 2024 symposium theme is Growing into New Tech, Plant Sciences in Bloom. We have gathered speakers from various backgrounds that research and work with technologies in many diverse fields. We hope to expose you all to the innovative work being done in plant and agricultural technology, the direction that it's headed, and how it can be applied to the various fields of plant science. So without further delay, I'll pass it off to Joseph Walston, the current botany representative, who will be introducing our first speaker.
Thank you, Audrey. I am pleased to introduce our first speaker, Nate Kingsley. Nate Kingsley serves as the Seed Conservation Lab Manager at Lyon Arboretum in Honolulu, Hawaii. Before joining Lyon, he worked at the University of Tennessee, conducting phylogenetic analysis to reconstruct species relationships and taxonomic distinctions of bryophytes, fungi, and vascular plants. He also served as the curation manager at the University of the Tennessee Herbarium, focusing on digitizing the plant collection and modernizing the seed collection. His research interests include population ecology, plant phylogenetic relationships, seed storage behavior and dormancy, and native plant development for conservation. Specifically, he is interested in how biotic and abiotic factors shape plant population dynamics phenology, and their responses to disturbances, examining the evolutionary convergence of recalcitrant or desiccation-sensitive seeds of endemic Hawaiian species and how the production and function of plant hormones regulate seed physiology. Additionally, he is interested in utilizing seed morphology data to trace evolutionary history. Elucidating Hawaii's broad patterns of divergence and connectivity requires parsing the genetic implications of seed phenotypes en masse from large databases. An expansive new trove of Hawaiian seed morphology data developed by his team at the Lyon Arboretum and Seed Conservation Lab meets this need, uncovering emergent phylogenies across hundreds of species by integrating visual patterns with taxonomic context via computational methods. Please join me in welcoming Nate Kingsley to the stage.
Thank you so much for that introduction, Joseph. I'm Nate, and I'm the manager of the Seed Lab at Lyon Arboretum. Today, I'd like to talk about a little bit about the seed bank and also some projects that we're working on. So just a background, I work at the University of Hawaii's Lyon Arboretum. It's a 194-acre botanical garden in the back of Mānoa Valley on the island of Oahu. We have a palmedum there as well, and of course the seed lab. How do I use the? Oh, cool. The seed lab and the micropropagation lab. So these two labs actually comprise the Hawaiian Rare Plant Program, which was developed to address a critical need of the rare plants that we have in Hawaii. It was founded in 1992, and with an 89% rate of endemism. There was a lot of isolation events that made it to where a lot of these species were critically becoming endangered rapidly. And so we're also working on developing a cryopreservation program, and a lot of our plants that have come out of the seed lab and the microprop facility will then go to our rare plant nursery. So this illustrates this critical need. So out of the 925 species that are federally listed under the U.S. Fish and Wildlife Endangered Species Act, 429 of them are in Hawaii alone. So this is about almost 45%. This next graph also illustrates this by the number of threatened and endangered species per 100 square miles by region. And in Hawaii, it's almost 4. We can see it's like drastically different than a lot of these other areas. And therefore, Hawaii's been referred to as the endangered capital of the world. So a little bit about the way our workflow works is we partner with the state, the state Division of Land and Natural Resources, that will go do surveys to map out these rare plant collections, make the collections, and then they will go to the rare plant facility where we'll then decide if they should be go to the seed lab, the micropropagation facility, or both. We're working on developing our cryo program. So right now we just have the 2 doers where we're primarily trying to focus on Petrardia, as the coconut rhinoceros beetle has become a looming threat, wiping out a lot of our endemic palm. And then after that, they'll go to be hardened off at the greenhouse and then used for restoration outplantings in the future. Last year, I think we wrapped up the year with sending out over 2,500 critically endangered species that were used in outplanting. So the Seed Conservation Lab is the largest seed lab in the state, and it's the endangered repository. So all the endangered collections that are made on all the other islands, not just Oahu, will come to us and be banked at Lyon. We have, this figure's a little old, I think we're closer to 34 million now, with almost 700 native Hawaiian taxa, with about 70% of them being endangered species. So we've got a lot of work that we're doing in the lab. A little bit about some of our research is we're trying to conduct storage trials and viability tests to understand the storage behavior of a lot of these species. The rate of recalcitrance, I'll start here. So where the water removal is lethal and the seeds can't be dry, they can't tolerate desiccation, therefore we can't store them in seed banks. It's typically higher in tropical environments. And so early on when the seed lab was originally founded, it was kind of this trial and error with this expectation that maybe some of these wouldn't be able to be stored. Luckily, though, we found that the rate of orthodoxy in our collections was almost 70%. Those are ones that can tolerate drying and freezing, and which is the most cost-effective, resource-effective way to store seeds long-term. And that's really good news for us. And so we're able to store these in cold conditions. And so far, we have a little over 30 years of data, and with some species maintaining within 70% of their highest or initial viability. Hawaii is interesting in the case of its level of intermediate taxa, where some of these species, you can remove the water, but they're temperature sensitive. We used to say freeze sensitive. We've since changed that. Temperature sensitive at conventional -20. And we changed that because we suggest that you might be able to store these at -80. We're still trying to work on some of the research of that now. Intermediate taxa can also include those that are short-lived, and so the viability will decline approaching 0 within 5 years. We think that this sensitivity at -20 is due to like a lipid thermal melting event that's going on with a higher oil content in the seeds. Some of the other work that we're doing is working on trying to break dormancy to optimize germination in a lot of our species. Some are really complex. I won't go into this in detail, but recently we had some species in the Rutaceae. We have the genus Melicope. It's an indigenous genus, but all 56 taxa within Melicope that we have in Hawaii are endemic species. It was actually previously thought that it couldn't be stored, that it might be toting the line between intermediate and recalcitrant. But we actually found that it was a complex dormancy, that we actually use fire and gibberellic acid to finally break this dormancy after all these years. And so now we're pulling these out to try to see if they actually can tolerate freezing, and then we can re-describe what those storage behaviors would be. As well as routine viability testing. So as needed, we'll pull these seeds out of storage. We always try to have an initial viability test going, so we have a baseline or a threshold that we can hold the subsequent viability test to. Recently, we acquired an X-ray that we can use to kind of assess the potential to germinate by looking at the fill of embryo in the seeds, and then actually run those viability tests for each species. Those that are pulled out will be grown out in our incubator, and then those are the ones that are used for outplanting later on. We do this in order to assess longevity of these species. So seeds in storage, seeds pulled out of storage, the viability never improves. It is always declining or somewhat sustained, but it's never improving. But for a lot of these species, it's a gradual decline. And so that's what we want to monitor. So we pull them out every 1 year, 2 year, 5 year, 10 years, so on, where the data is needed to try to understand how long we can best keep these, and then describe recollection intervals for these species to make sure that we don't lose individual founders. Each collection that we have is tracked to its specific location of collection, as well as the maternal line that it was collected from. So we want to track all of those, and if a specific maternal line is declining and that founder is still there, we want to make sure that we can tell our team so that they can go out and collect it. This is important because a lot of the species we work with, like, yes, I mentioned that they're endangered, but we also have a localized conservation status that is referred to as PEP, PEPP. It's a plant extinction prevention program that goes out and defines these that are that have this PEP status as less than 50 individuals in the wild. And for a lot of these, that 50 is on the higher end, and we're working with species that are usually less than 10. So it's really important that we keep track of these. In recent years, we've really developed our partnership. It's grown significantly in the past few years. Even when I started, there were only about 7 or 8 people that would attend these meetings, and back in August at our last meeting, there were about 45. So that's great. And a lot of these smaller short-term restorations seed banks for common species have been popping up, and they can work with us really well to help refine our conservation efforts. While we focus on long-term conservation, they can support spreading natives and creating this native seed supply chain of common species. This is a cool infographic that made last year. All right. So one of the projects I want to talk about was this digital database of Hawaiian seeds. I want to just point out, I like this slide. It was a title slide when I'd given this before, and it's older. So some of these pictures, just pay attention to them now. These are some of the older photos that we took. I'm going to address that again in a minute, but just remember that. So in 2020, we were deciding how to address this need, where we understood that Hawaii is an extremely isolated archipelago, and even within the state, we have to take planes to get to each other on different islands. If all the endangered species and some common species are coming to us at Lyon, if you don't specifically live on Oahu, it's very difficult to access them. And even more so, even with access, it's not great for the collections to pull these germplasm out as they warm up post cold storage and then retain moisture. Viability starts to decline. So what we wanted to do was to try to broaden this access, as well as for mainland researchers like even some of you in the room that I've spoken to earlier today and yesterday, that have been working with Hawaiian flora or Hawaiian seeds. We wanted to make this accessible to everybody, improve the identification of these species by having these really high resolution images, and then also help with any kind of research by collecting this data that maybe somebody that was visiting would want to collect. So we developed this system of stacking these on the Z-plane, multifocal stacking, about 8 to 40 photos for each specimen, where we're trying to think about the things like the size, the shadow, the orientation, the background of these, and how we can best make sure that we're capturing as much morphological features as we can in a single image. So originally when we were doing this, we had a camera insert that I could put into one of the lenses of a microscope, as well as a 3D printed holder thing for your phone. And we would manually fine focus each point on the Z-plane and take those and then compress those together in Photoshop. We did automate a script to do it in Photoshop, so it made it a little bit easier, and then process those images in Lightroom. I keep this slide up because we've since moved from this method, but we're working on developing a protocol that makes this accessible to anybody, regardless of funding or any other limitation, that you can do something like this with what you have in-house, and it's better to have something than have nothing. This was one of the species I had, as well as some of the other ones from the first slide. This was one that I had sent to IMLS after we did this with our 3D printed holder, or this random camera that we found. Okay, hey, this is what we can do. We did this by manually stacking these things. And they were like, okay, yeah, that's great. What else can you do? And lucky enough, we were funded. And so we bought new equipment that we could use to actually automate this process with a Leica stereoscope and this Leica software. But it was helpful to have had done this manually, so that we understood what we needed to capture within these focal planes, like what specific aspects of the seed, what characteristics of the seed would change those images, and how to best adjust that. Because even here, you still have to specify the range and the stacking. Oh, good, it's on the photo. Yeah. So that really helped us. This is the same species that I showed before. And this was the kind of picture that we could take once we refined our technique and improved some of our camera equipment. And it looks drastically different. But I kept up the old slide too to show you, though, that some of those were great, and especially with the less textured ones, they were easy to do with what we had also. Just another example of the Z-stacking. If we were to take it on one focal plane, right, this is what it would kind of look like, and this is all we could get, especially this up close to the seed. These are very small seeds. But with our Z-stacking technique, we can put those things together, we can compress those images and encompass the entire depth of field and really improve the output. And you say, okay, I still don't believe you, Nate. I've got another one. So same thing, right? Blurry here, and then here we can kind of get the entire seed in one image. And so these are great. So they started looking like great photos. So we got that down. We knew what we wanted to do. And so we took 3 photos for each taxon. We have the close-up so you can capture any minute morphological detail in those. A dorsal, ventral, or proximal, distal, depending on the shape and size of the seed. That way you could capture different axes. And then a cluster for any morphological variation in the seed. This one was good for this photo. The cluster, not so much. But we have some other ones that they're very different within those clusters. We worked with our other partners at Bishop Museum to take images of the large seeds that wouldn't fit under our microscope. And so this still required that manual stacking since we had to do it with this digital camera and there was no software. But it was great because we knew how to do it. And so we could still get a really nice image of something that it's a very, very large seed. So then we have all these photos and we're thinking, okay, what do we do with them? And so we developed this website, seedsofhawaii.org. It is up. You can go there, where we house all of our images. And this was great because it kind of gave us the flexibility. Originally, we were going to put it onto a Pacific Consortia website that I later found out that UH no longer had the password to. Yeah, it was shocking. But this was great because, it required a little bit more work, but it gave us a lot more control. It's not hosted on the University of Hawaii's server, which gave us even more flexibility to do what we needed to do. And we uploaded all the images onto here, and now you can go up there. So we have, if you were to click on a certain image, it would take you to an individual landing page for each species. I put this one up here because I know everybody likes this one. So here we have the Hawaiian name, family name, some of the trait data that we've captured for the species, as well as the description from the Smithsonian Manual of Hawaiian Flora. And its federal listing, its dispersal, all that stuff. We also tried to incorporate advanced search features. You can search by island. Now, the caveat here is that a lot of these are endangered species, so true location information is obscured, but it does have island distribution or like island endemism that you can track by, as well as like an advanced search here, where similarly you can do that, but you can also search by what specific family you're looking for, length, mass, or something if you're unsure about the ID of that species. So we had this. And then to take that even a step further, we thought, okay, what can we do to make it to where we can increase the utility of the site and improve our searchability further? And so we wanted to incorporate the use of large language models so that we could improve the search process here and process the data in a way that will make connections and improve the searchability. So here we look at the predictability of these large language models, where they're developed in a way where they're predicting the next word or the next sentence or the next paragraph, this distribution of what would come next. But where we would run into a challenge here is it's by itself not great for scientific data. We have image data, we have text data, we also have numerical data. And so this would pose a little bit of a challenge just using the base model alone. So to refine that, we are looking into a retrieval augmented generation system that we can use to use the data that we have to sit on top of a pre-existing AI engine to kind of direct the data to, so that a query is directed directly to the RAG system that we're feeding in. It'll help with improving the accuracy of what's being queried. Oh, forgot about those guys. So here you can see the data that we have that we're feeding into our system is sitting on top of one of these pre-existing models. In this case, we'll maybe start with OpenAI. We can feed it directly into our vector database through embeddings. Here you have these proprietary platforms that can kind of serve as a middle between the two. and you can most certainly use them. But what we've found is that by doing that, you don't always see how your data is being embedded into your vector. So this illustrates that point further, where here within a platform like Chatbase, we can see the URL that's being scraped, but we don't know exactly what the data is that's being scraped. And it may be an incompleteness of data or an accuracy issue. So what we wanted to do is then find a way to directly embed these into our vector and bypass these proprietary platforms so that we can sit this directly on top of this engine, having full control over our data. And so we did that in an organized JSON schema structure, detailing exactly what fields are expected and how these values are represented. And so you can see here where we scrape directly from the front end of the site. We're pulling those, sorry, there's a lot of lines, but you see they match to each other. And this way we can see exactly what's going on, what exactly we're pulling, define what we're pulling, and then use those, and it'll generate a TXT file output that we can then cross-check again. So now this TXT file is here on the right. We can check again, make sure that it is what should be pulled. We have accurate data here, and then check it against as we create our embedding. And this gives us like full control over the data that's being pulled and then embedded into our vector database, so we know exactly what's going on. And that way we can make meaningful connections within our vector database here. So these vector databases are specialized storage systems designed to handle and query this vector data. It's a format that's ideal for AI. The closer that these things are related in this hyperdimensional space, the closer the thing that they are, the more related they are, and the further away, the less related they are. And just one note, it's important to understand that the different types of data that you have may require different models. So by having something like this JSON schema, we have the flexibility and the adaptability to move between models because it's the data that we've defined and pulled, and we're not stuck to a single platform. And so we can use different things like OpenAI for our text and Tabular Transformer for our mass data, and then Gemini for our image recognition of the vision of the images that we're uploading. So some specific examples of how this would apply to our data. We can use this to try to understand texture. We can run these kinds of models to see like this kind of pre-processing so that we can find the most effective approach to get the most accurate output for something like seed texture, where if it maybe has a lot of ridges and valleys, we can use a Fourier transform, or if there's a lot of roughness or hairs, we can use something like a fractal dimension. And similarly with shape, we can use some of this pre-processing by like using these predefined geometric models and then using the output of what we get from a script where we're trying to understand contour and seed shape and use those to make inferences and compare these shapes within our model. So here we're looking for like even extreme values. So like the low circularity caused by this extreme divot is indicative of this cardioid shape. Or the really high eccentricity through this elongation is this oblong kind of ellipse. And we can then take the masks that are produced from those, these are the same, Silene and Gaunia, and we can define, okay, this is the inner bound of this cardioid shape. This is the inner bound of this elliptic shape as we fully understand it, given those extreme low and extreme high values, respectively. And then parse and compare similarities and differences. Shadea, Pentandra, Silene, they're two genera, both in Caryophyllaceae. Also a cardioid shape. Having those in the same inner bound. Claremontia, more of an ovate, but still oblong, maybe within that outer bound, but not quite an ellipse. And we have that, and we can make this in this abstract hyperdimensional space, where again the Shadea and the Silene are closely related, and these are further apart and even further apart from these. And as we start making these kinds of connections, we can better understand the connectivity of our data and how we can improve output depending on what's being queried by the user. What do you want to see? What questions do you have? Whether it's for species identification, you want to see different seed sizes depending on rainfall or location, you can do that. And we wanted to make this very dynamic and engaging for the user. We documented a lot of this in our original small article where we talked about how someone can do this, whether it's little funding or a lot of funding. And we plan on continuing this and putting together these resources so that they can be openly accessible on our website and the code on GitHub so that others can do the same thing and follow a similar model. Where we kind of want to take this going forward is continue to add this trait data and really finalize these resource guides. Imaging the non-native species is going to be a big one. We've had some conversations with USDA recently about the great need to have something like this for noxious weed biosecurity at Hawaii's borders. It's become a real issue. And we don't really have the team needed. And with something like this, we could hire more generalists within USDA and use the machine learning and have those that are hired as kind of checking to make sure that the machine is generating accurate responses. And since we already have the tools that we need to develop this, all we need to do is just get more of these non-native taxa. We, of course, don't bank invasive species. Fruit and seedling photos. That one's been a big request from our collaborators and our colleagues. So we want to do it. I think it's going to take a while, but we're going to try. As well as the X-ray images, so we can kind of show an idea of like some of these that have low fill and give our collectors an idea of these are the species that typically we should target to make greater collections of. We have a real problem with this from our silver swords up at Haleakala, and continue to refine this AI model. I'd like to just thank a lot of our partners and collaborators that have helped with our collections. A lot of other seed banks have been super generous. When we imaged everything in our collection, I reached out to all the other seed banks, and they've been super helpful in sending us a lot of common species that we didn't have. As well as IMLS for kind of believing in our project and funding something like this. It's been a huge help, and we've made it really far, so it's been great. And with that, thank you all so much, and I'll take any questions if you have them.
Thank you, Nate. Before we hit the Slido, does anybody have questions they'd like to raise their hands for now? Great, yeah.
That was super cool. Is that better? Yeah, amazing talk. Thank you so much. I'm curious with the conservation efforts as you're collecting the seed and you have this like really amazing repository of data on the morphology. Are you tracking how that's changing during the process of outplanting and cultivating those collected populations? Kind of curious about like indirect selection for seed production of these species and whether it seems like you have a really nice opportunity to study that with the wealth of data that you're collecting.
Yeah, absolutely, and we totally can, and we have all that up there. I think the only caveat with some of these is like the extremely rare ones, where we're maybe working with one individual, and sometimes that is, I hesitate to use the word bottleneck, but a very conservative bottleneck where if we're working with a lot of species that have 5 or less individuals, they usually will stay in the greenhouse for quite a bit of time until we can get more seeds from them there. But then yes, then we'll take that and process that data and compare.
Thank you. That was a great talk, great work. And I have a question with the tools that you've been using. Would you be able to use this also for like in terms of identification for fossil and subfossil seeds, do you think?
Yes,
chart?
I think so. I had I'm forgetting her name. We had just gotten contacted about that actually from a person at USGS. She sent the seeds, and then I came here, so I don't have them yet. But they pulled a bunch of seeds from a core around the north shore of Oahu, and they're in rough shape. But I'd like to try to see if we can use some of the features of this model to see if we can identify them.
Yeah, there are some seeds here in the lab that are in better shape, maybe.
Okay,
we
we'll
can
take a look
talk
then, yeah?
about. Okay, thank you.
Thank you.
Any other questions from the audience? Perfect.
How do you actually find these founders? Like finding one plant that potentially no one's seen before in a jungle seems impossible.
Yeah, it's an undertaking. So again, partnering with the state, it's not a huge team. I think in total there's like 8 people on each island, give or take. They kind of go out and do these massive surveys, and it's been decades of tracking. And sometimes it's serendipitous, just coming across them. But yeah, we have an extensive mapping feature, and we have a lot of like historical populations. So we work pretty heavy with them, as well as like Bishop. We have a lot of like historical. They have the largest herbarium in Hawaii, and we work with them. We have some like historical population records and stuff too. So we're working now actually to try to combine all of our databases. This is independent of this project. Our working databases that actually has a lot of this raw data, so that it's a lot easier than us trying to email back and forth. But that's the way we do it right now.
We have some questions over here. First, if a seed is no longer viable, is it still worthwhile to image it?
Oh, yeah. Yeah, I'm sure there are several that we've imaged. I think some even in the slide. I had some, I think the first one where I had the reasons why you would image your collection, that Dubaudia, they're empty. Those are some of the silver swords, and they're not viable. As a matter of fact, they're pretty empty. The understanding of that though is that's where we would make adjustments, because I won't take the weights of the empty seeds, obviously, right? But the picture itself is fine. If there's any reason that we believe that something isn't image worthy, as it's not like effectively characterizing that species, we'll try to find another, and we have a lot of checks in place to make sure that we don't do that. Shriveled seeds, immature seeds, things like that. But we have, in terms of immaturity, we actually have kept those because that's another thing that we'd like to eventually do with this system, is have a way to assess maturity based on like seed color and seed size, which would be super helpful for our collectors. We can't bank immature seeds, and so it'd be a great way for them to know, so we don't keep receiving them.
Awesome, thank you. And we also have a question on Slido. How do you cryopreserve micro-propagated plants, and what is the percent of viability of these plants after X amount of time?
Okay, I don't do this, so I don't know for sure. I know with Petrardia, the endemic palm, that they're excising the embryo and treating it with cryoprotectants. I don't know that. I guess they could be pulling shoot tips from micropropagated plants, but I'm not 100% sure if they're doing that. And theoretically, the viability, although it depends on the species, so I can't give a number, but theoretically the viability would be sustained greatly in cryopreservation. As it's so cold, you've essentially halted all metabolic processes, so you wouldn't really see much of a decline.
Another question that we have is, do you ever have seeds so small that light microscopy could not capture the details that you need? And if so, could like electron microscopy work with your pipeline?
Can I go back? This is the smallest seed that we have. This is an orchid. And so we were able to get, it looks pretty good. It's interesting, it's so small and translucent, you can see the embryo inside. So we were able to capture that. I know that I think they had done some like orchid imaging or viability imaging with fluorescein diacetate at Q. but we don't have that. But luckily, we were able to image our smallest seed. We only have 3 orchids, and this is by far the smallest. So.
Okay, thank you to everyone for the questions. If we didn't get to yours, or if you think of one later, please be sure to save it for the speaker panel this afternoon. Let's give another thanks for Nate for coming today to present for us.
Thank you.
Now, John Searle, current Vice President, will introduce our next speaker.
All right, thank you, Joseph. It is my honor to be introducing our next speaker, Dr. Joe Gage. Dr. Gage is an assistant professor at North Carolina State University in the Department of Crop and Soil Sciences. Dr. Gage's research program is focused on linking crop genetics and phenomics to understand how to develop more resilient and productive crop varieties. Current projects include studying how sequence variation controls gene regulation, how gene regulation contributes to genotype by environment interactions, and how novel methods for processing and interpreting high-throughput phenotyping data. Before we welcome Joe to the stage, there are 2 points I would really like to bring up. Is Dr. Gage is a proud UW alumni. In 2018, Dr. Gage graduated from here with a master's in biometry and biometrics and a PhD in plant breeding and plant genetics. And I would also like to mention that in 2015, Joe likewise served as the Vice President of PSGC. So with that all, please welcome Dr. Joe Gage back to UW-Madison.
Thanks, John. That was a really nice introduction. It is it's super fun to be back here, and probably a lot less nerve-wracking to be up on this stage giving a talk instead of organizing this whole event. And the hospitality has been fantastic. I think the students have done a really good job putting this all together. And so, yeah, it's fantastic to be here and to be a part of this. Okay, yeah, so as John said, I'm at North Carolina State University, and my group focuses on using high-throughput phenotyping as well as molecular assays of gene expression to understand how crops perform well in particular environments or don't. So I want to get out ahead of things and sort of address the theme of the symposium today, growing into new tech. My program uses a number of different technologies. Some of them are newer than others. But for the ones that aren't particularly bleeding edge anymore, we are thinking about ways to apply them at new scales that they haven't been used before. So if it's not new, we're growing it. In addition to some of the technologies that you see on the screen here, though, I also want to sort of point out our use of another new form of technology that's really making inroads everywhere, and we just heard about it in our previous talk, and that's sort of the application of machine learning and AI to biological disciplines. And so keep an eye out for this Happy Mac logo in the center here. That'll show you sort of places that we're using machine learning in our research. So I want to start off by giving some sort of rationale for why we study what we do. The United Nations publishes these Sustainable Development Goals, which are 17 goals that serve as a blueprint for greater equality and prosperity across the globe. And the vision, or my vision, which is sort of how I see a better world, is that we have resilient, adaptable, and sustainable agricultural systems. And when we compare that vision back to these development goals, having improved agricultural systems can at least contribute to the accomplishment of at least 12 of the 17. So ag is really fundamental. In addition, the World Economic Forum publishes this global risks landscape, which shows on the x-axis the likelihood of particular risks, and on the y-axis, the potential impact of those risks. And you don't have to be able to read the actual labels on the dots, but I want to point your attention to all these green dots in the upper right-hand corner. So high probability of occurring and high potential impact if they do occur. And all of these are related to extreme weather events, biodiversity loss, and climate change. And so these also, I think, really underscore our need for developing resilient agricultural systems that can weather the storm, so to speak. So the mission of my research group, which is sort of how I see us supporting this vision, is to better understand and predict how particular genetics will perform in particular environments. And this is really important because, say you are a plant breeder and you develop a variety that performs wonderfully in Wisconsin, there's absolutely no guarantee that it'll do well in North Carolina. And this is also particularly relevant in the face of climate change because it takes quite a bit of time to develop new varieties. And if you are breeding for the climate of today, even in Wisconsin, with the hope of producing something that can be grown and harvested in 10 to 15 years, there's no guarantee that that's going to work either. So there's a certain amount of fortune telling and looking into the future that goes into developing productive varieties. And so my group does research to support this mission in sort of two distinct but complementary buckets. The first is thinking about sort of what I term macro plant level responses to the environment. You can sort of think of this as like your traditional like high throughput phenotyping. The other region that we do research in is studying how plants regulate their gene expression in response to cues that they're getting from their environment. I'm going to talk about both of these today, but I'm going to start off with high throughput phenotyping. So we are in a particularly exciting time to do high throughput phenotyping, especially because equipment and the computing power to process data coming off of that equipment is fairly affordable. And this leads to, or gives us the ability to generate mountains of raw data. The crux of this, though, is sort of how to get from mountains of raw data to useful information. And so the sort of difficulty of this question poses a couple of roadblocks between our research interests and our mission, which are first that in order to effectively use plant phenotype data to understand how different genetics perform in different environments, we need lots of data to build robust models. And generating that data is not easy because it's difficult to efficiently extract information from raw images or other forms of data streams. So one way to go from raw data to useful information is to build models to predict or measure traits that we actually care about. I'm going to give a couple examples of how our group is doing this right now. So first has to do with canopy structure in maize. So having maize plants with particularly upright leaves near the top and sort of more horizontal leaves near the middle and bottom can really help the plant maximize its interception of sunlight and maximize photosynthesis throughout the canopy. And there have been a couple of nice papers published this year identifying and characterizing genes that contribute to this canopy architecture. But in addition to single plant canopy architecture, it's also important to think about how plants grow in relation to their neighbors. So by sending leaves into the interrow space, plants can maximize on available space and sunlight sort of cooperatively without competing with each other. And so in the bottom left here, we have an example of plants that are growing their leaves perpendicular to the row they're planted in and parallel to each other. And on the right, we have an image of plants that seem to be growing in sort of random orientations. And as a result of this, their leaves may be blocking the leaves of their neighbors. And you can imagine as a whole, the canopy may not be intercepting as much sunlight as it possibly could. And so Yan Zhou at Iowa State University has done some really nice work characterizing this leaf orientation and shown that there is a genetic basis for the way that plants orient their leaves relative to each other. The issue with this is that it took quite a bit of manual annotation of images in order for Yan to do this study. And so in collaboration with Weilong He, who's a PhD student in the biological and ag engineering department, we've developed a model that can take images, identify the leaves in them, and quantify their orientation relative to the row that they're planted in. And this is really exciting because it allows us to start thinking about canopy architecture and leaf orientation in large populations, across environments, and throughout developmental time. So as corn plants come up, they have all the space in the world. As they get larger and larger, they start invading on each other's airspace and need to orient themselves in order to maximize efficiency. So we've got the model developed. We're still working on applying it to gain biological insight, but just as sort of a nice proof of concept, we see that when we do GWAS on our estimations of leaf orientation, we hit some of the same genes that were shown on the previous slide. So another example is Cole Hammett's master's thesis. So Cole is a plant pathology student who works on foliar diseases in maize, specifically southern leaf blight. And scoring foliar diseases is extremely time-consuming because you have to walk the field and score every single plot on a scale from 1 to 9 for how bad the disease is. And in addition, it requires a certain amount of expertise. So you can't just send an army of people into the field to do this. And even if you do have the expertise, there's a considerable amount of bias between expert scorers, which overall makes quantifying disease severity fairly difficult. So Cole is collecting tens of terabytes of data on large populations through time with the intent of building machine learning models to quantify disease severity both faster and with lower bias, hopefully depending on our training data, and again with higher temporal resolution. So it's a couple quick examples of building models to predict traits we care about most, but the alternative way to get from raw data to information is to just let AI decide what the traits are that we're measuring. And so to do this, we're using a machine learning architecture called an autoencoder, which you can think of as basically like a fancy image compression algorithm. So we start with something very high dimensional, say an image of a corn plant, that may have tens of millions of pixel values. And the model compresses that image down to something that's called a latent representation that has on the order of tens to hundreds of values instead of tens of millions. But the trick here is that the model learns to do this in such a way that that the latent space representation is as information rich as possible so that the original image can be reconstructed from those values, at least as well as possible. There's always at least some loss, and so that's supposed to be demonstrated by my slightly fuzzy corn plant on the right-hand side. But we can treat the values that come out of this latent representation of these images as phenotypes themselves. And these have a number of advantages over sort of traditional manually measured traits. The first is that machine learning models are particularly adept at distilling useful information from noisy sources. They have the potential to minimize human bias in deciding what we're paying attention to. And just had a conversation with somebody about this morning. They also reduce the considerable amount of effort that goes into building bespoke models to measure a particular suite of traits in a particular system, which is something you can spend an entire graduate career on. And then finally, they allow high volume characterization of plant phenotypes. And so this has the potential to address the two roadblocks that I mentioned earlier, in that they can facilitate us efficiently extracting information from raw data streams, thereby creating lots of useful data that we can use to build models and understand how different genetics perform in different environments. Okay, so our first attempt at doing this, actually instead of using images, we used LiDAR point clouds that were collected. So this is a 3D representation of a single plot of maize collected from a LiDAR sensor that's mounted on a robot that drives through the field. And we fit the model, okay. Question is, are these latent phenotypes telling us anything useful at all? And we used a couple of metrics to try to get a feel for this. The first is heritability. And so heritability is a number between 0 and 1 that estimates the amount or the proportion of variance in the phenotype that can be attributed to differences between genetically distinct individuals in your experiment. And just as a baseline, for manually measured phenotypes in this same experiment, we saw heritabilities ranging between about 0.25 and 0.8, depending on the trait. And these latent phenotypes had heritabilities that max out in the range of 0.25 to 0.4. So they're demonstrating levels of heritability similar to medium to low heritability manual measured traits. The second question that we asked was whether there's sort of any useful biological information in these traits. So we asked whether we can predict traits that we would normally measure by hand from latent traits. And the answer here is also yes. We get fairly good correlations between predicting things like flowering time, plant height, and yield from latent traits to what we would get measuring them by hand. So the takeaway from this study was really that yes, latent phenotyping models do appear to be distilling some amount of biological information, but this was really kind of just proof of concept. And so moving forward, now I'm working with two PhD students at Cornell, Peter and Aaron, who are involved in, one, trying to improve model architecture and our training methods in order to make these models more efficient. Two, train them to utilize different data streams, such as multispectral imagery. And three, adapt them to longitudinally collected data, so data collected throughout the season that can help add a dimension of sort of temporal variation to these traits. Another thing that Peter and Aaron have been sort of really thoughtful about as they've worked on these models is thinking about baselines to compare against. And so one thing that they've started doing is comparing their models to NDVI. And NDVI is a vegetative index that's based on relative proportions of red and near infrared in an image. And NDVI has been shown sort of time and time again to be a decent predictor of kind of overall plant health. You can often, at least in maize, predict things like yield and flowering time from NDVI data. And so in these experiments, NDVI does a decent job of predicting things like flowering time, plant height, ear height, grain moisture, with sort of average correlations ranging between 0.2 and 0.3. When we compare instead their latent phenotypes to NDVI, we see much improved ability to predict these manually measured traits from the latent phenotypes. And so to me, this really indicates that these latent phenotypes are capturing additional information beyond sort of one of the community standards that can be useful for improving predictions of traits that we know that we care about. In addition, you'll remember I showed in our original study, latent traits had heritabilities about 0.4. through improvements to the model and the way that they're training it, Peter and Aaron have boosted that to ranging between about 0.3 and 0.8, which is fantastic. So looking forward, they're now kind of working, as I mentioned before, on integrating sensing modalities into a single model, as well as pulling in this kind of longitudinal aspect of data collection, which is super exciting. One shortcoming of these latent phenotypes is that they do sort of integrate signal over genetic effects and environmental effects. So you can imagine if you take a picture of a plot, and then you take a picture of the same plot 5 seconds later when the wind is blowing, those images are going to look different. And ideally, you would want your model to extract the same traits from both images. But as it stands right now, they largely, or not largely, but they'll extract sort of genetically relevant information as well as information about the leaves blowing around. So a nice recent study out of Iowa State and Nebraska has shown that by using making some clever changes to the model architecture, they can actually sort of separate those genetic and environmental effects, which is something that we're really excited about and looking forward to implementing. In addition, you might be skeptical about whether these traits are useful for biology. But we've also seen some nice work coming out of Nebraska where Michael Tross used hyperspectral data collected on leaves and then mapped his latent phenotypes against the genome, and was able to find hits to genes that are involved in leaf physiology. So really nice sort of proof of concept there. We're excited about this field moving forward and think that it holds a lot of promise. In particular, because using AI to, in a sort of unsupervised or semi-supervised manner to extract traits from high dimensional data means that we can start feeding these models large data sets from all sorts of disparate sensing modalities. So things like UAV images, hyperspectral reflectance, satellite images, 3D reconstruction, so on and so forth. Moving forward, I really see this sort of integration of differing data streams as a way to characterize plant phenotype at scale. And so one of the, as I mentioned before, sort of the crux of building models for characterizing plant health and form across lots of different environments is getting high quality data. And so we think that latent phenotypes have the ability for us to phenotype plants across environments and through time at scales that we really can't do manually. That's all I've got for plant phenotyping for today. I'm going to switch gears a little bit, talk about gene regulation. So we know that distinct individuals with distinct genetics have different profiles of gene expression, and that's what I'm showing in this figure here, which has shows gene expression. Each individual is a dot, or each dot is a gene. Shows differences in gene expression between two inbred lines. And the fact that there's a lot of deviation from this one-to-one relationship means that these individuals are expressing different genes at different levels. We also know that plants regulate their genes differently in response to environmental cues. So here we're looking at a number of genes that are either upregulated or downregulated in an inbred line called B73 in response to cold stress, I believe. But what's particularly cool, I think, the most exciting thing sort of about this area of study is that varieties that are genetically distinct from each other show differing responses to abiotic stress. So each of these rows is a genetically distinct individual, and we can see that the degree of up or down regulation of genes in response to cold stress differs. And if this is something we call genotype by environment interaction, or I'm going to just call it G by E from here on out. If we zoom in on, say, a single gene in two individuals, whoa, that's pixelated. we can see that the individual with the red allele, say this is B73, shows upregulation of gene expression in response to environmental change, but other alleles, say MO17, may remain constant in the face of the same environmental stimulus. And so most of the studies sort of looking at this up to this point have been done in controlled environment conditions, so growth chamber assays, small number of plants. We're really interested in how these patterns play out across space and across time in actual real-world field conditions. There's my pixelated picture again. Our sort of current working model of how this plays out is basically that changes in or variation between individuals, which may be genetic or epigenetic, leads to this sort of G by E for gene expression, which when you then accumulate it over tens to thousands of loci in the genome, causes organism level phenotypic G by E for quantitative traits. And so one way that we're starting to study this is looking at how transcript abundance changes in response to genetics and environment in real field conditions across the corn-growing regions of the United States. And so Ty Thomas is a PhD student in our group who is working with a sample of several hundred hybrids grown in about a dozen locations across the US to understand just how much variation we can attribute to genetic effects, environmental effects, and this G by E that I showed. To do this, basically just treating transcript abundance or gene expression like any other phenotype. So he's modeling transcript abundance as a function of some fixed effects that account for sort of nuisance parameters, but then genotype, environment, and G by E effects. And he fits this model 30,000 times, once to every gene in the genome. And from this can sort of get a 10,000 mile view of how genetics and environment and G by E are playing out in gene regulation. So in this figure, we're seeing distributions of variance attributable to each of those effects across 30,000 genes. And so some takeaways from this, first off, most of the variation goes into error, and this is totally expected, okay? Transcript abundance is really stochastic, and I think it is impossible to ever fully explain all of it perfectly. But we see that environmental differences are really important, the next leading cause of variation in gene expression levels. And we see that G by E is really rampant. I mean, there's some level of G by E for almost every single gene in the genome. And on average, it tends to play about the same degree, it has about the same role in gene expression as it does in whole plant phenotypes like plant height, flowering time, which is pretty cool. And so moving forward, Ty is really interested in sort of digging into what's causing this variation. So understanding, first off, how G by E for transcript abundance translates to G by E for traits that we care about, grain yield, right? But then also understanding what's going on at a molecular level. So I sort of posited that changes in gene sequence or epigenetics may be driving these differences. And so he's also thinking about how to figure out what's driving G by E for gene expression and how we can use that to breed more resilient varieties. In addition to studying gene expression across environments, we're also looking at how gene expression changes across time among genetically distinct individuals in the same environment. So we know already that many genes show oscillation in their level of expression over the course of a day or 24-hour period. And sort of have an example of that up here. Depending on sort of how you define this and which systems you're looking in, this can be as many as a third of genes in the genome. When we look in maize across genetically distinct individuals, what's really interesting is that we see that the number of genes that show this sort of rhythmicity over the course of the day varies almost twofold among diverse genetic inbreds. So the fact that there's genetic variation for this rhythmicity of gene expression throughout the day led us to build models to try to explain or understand what's causing that variation. So we're building machine learning models that take as an input the promoter sequence around the transcriptional start site of each gene. And the goal of the model is to predict whether that gene shows cyclical expression throughout the day or not. There's a picture of the model architecture. After we train this model, the fun part is that we can kind of peel back the top and look at the sequence motifs that the model is identifying as important for predicting whether a gene is cyclically regulated or not. And when we do that and look at the motifs that this model is paying attention to, we find some interesting things. Here's an example of a motif that the model thinks is particularly important: AAA, TATCT. This is an absolute dead ringer for the evening element, or the binding motif for a really well-characterized core player in circadian rhythm that's been characterized in Arabidopsis and other species. That's not the only one, though. We identify a number of these binding motifs that have already been discovered, characterized, validated molecularly in model systems. So what? You discovered a bunch of stuff that people have already discovered. What's cool about this, though, is that we're able to identify these motifs through training and investigation of a single model, whereas identifying all of these and publishing papers about them has taken dozens of years and extensive hard work in model organisms. So really great potential for using machine learning models to understand how sequence is regulating gene expression in your organism of choice. We're also interested not only in using patterns of gene expression to understand how sequence drives gene expression, but also to understand how patterns of gene expression drive adaptation to new environments. So just as an example, in this same data set, we find a number of genes where the haplotype of the promoter sequence for the gene is strongly correlated to or predictive of patterns of expression throughout the day. And so in this example, individuals with the purple sequence in front of their genes show oscillation over the course of the day, whereas individuals with the blue promoter sequence are just sort of constitutively expressed at a low level. So very clear differences in expression patterns. There are several hundred of these throughout the genome, and when we ask what they all are, find that they are enriched for function in the adaptation of maize from central Mexico, where day lengths are short, to northern latitudes with longer days, where most of maize is grown today. So really cool evidence for how changes in transcriptional regulation can drive adaptation to new environments. This is something that we're thinking about moving forward as a technique for adapting maize to new and changing conditions. To sort of further our work in that arena, we are currently just kicking off. We're about a year into a new study that we're involved in, characterizing genetic and phenotypic variation in heirloom varieties of maize from across the United States and Canada. So we have a really fantastic collection of diversity here that we're evaluating out in the field. Most of this work is being done by two PhD students, Jordan Cummings, who some of you may recognize. She did her undergrad here, and Melissa Draves, who's at the University of Missouri. And the hope here is that with this population, we can identify genetic variation in alleles that are involved in adaptation to the broad variety of climates that we see across the United States. So adaptation to cold, to heat, drought, biotic stresses, and use those to sort of inform or assist in adapting maize varieties to climates of the future. So that's it for gene regulation. Hopefully now you can sort of see how in tandem we're looking both at plant phenotyping and whole plant phenotyping and molecular phenotypes, both as a way to sort of understand and predict how different genetics will perform across environments, and thinking about how that will contribute to sustainable food systems in the future. Looking sort of into the future of how my program is going to continue to operate, it's becoming very clear to me that we're building really rich, large data sets in both of these two buckets, and that the time is really ripe to start combining those rather than keeping them separate. So speaking of modeling, I have to take this opportunity before I get off the stage to just give a shameless plug for the Genomes to Fields Genotype by Environment Prediction Contest. Would encourage you, if you're at all interested in modeling or predicting crop performance, to throw your hat in the ring. The idea here is if you sign up, first off, the competition goes live today, but you have until the middle of December to sign up. You'll be given access to phenotypic data from hundreds of thousands of hybrid maize plots across the United States from the last 10 years, as well as corresponding genotypic information for all of those, weather covariates, and your challenge is to predict how a set of unobserved genetics performed this past year in the field. And so if you win, if you do the best job, you get a bunch of money, you get a speaking invitation, some travel funding, and a guaranteed slot on a publication. So, and if you don't win, you'll have lots of fun and learn something along the way. So that's all I've got. I'm going to wrap up. Oh, red boxes to remind me just to say my lab is fairly young, and so we've got a lot of new and exciting things going on, and I wasn't able to talk about all of them, but I'm going to be here all day, and I love talking about the stuff that people in my group are doing. So if you're interested in microbiome, transcriptomics, nutrient use efficiency, allele-specific expression, translational regulation, or developing genomic resources for breeding, we're doing all those things, and I would love to chat about them. With that, I have to thank all of the people on this slide. They did the work, not me, and thank my funding sources as well. So with that, happy to take any questions.
Thank you, Doc. Oh, there we go. Thank you, Dr. Gage. We're going to be opening the floor up for questions. If you have questions, please raise your hand and have a mic be passed to you, or you can submit them with the QR codes that are on your tables with Slido. While you guys are getting ready, I have a question that I want to lead off for Joe.
All right.
So as plant breeders, we think of complex traits as being built up with many small effects. You talked about the machine learning approach of identifying the promoter motifs. How do you think that could be used for using machine learning to identify motifs for targeted editing to break out of this infinitesimal model that we currently live under?
That's a fantastic question. And I think that maybe the answer there is not so much in motif identification or the sorts of examples that I've shown today, but more so in some of these nice new like transformer or large language models that are identifying bases that are particularly relevant or important or conserved and important for function of genes that are known to be involved in traits of interest. And so I think sort of using more of a evolutionarily guided approach may sort of yield information about, how edits to your gene of interest may have been tolerated or not tolerated across evolutionary time, and that there has been some really nice work on identifying functional variation in coding regions and regulatory regions that would probably be relevant.
Awesome. Thank you very much. Have we handed? There we go. We'll get some Nick.
Hello. Thanks for the talk, Joe. I liked your point about AI moving us forward and looking back at the Arabidopsis models and how they confirm the results. What happens when we start moving too fast? In other words, have you put any time into thinking out strategies for verifying and how many of the hypotheses we're going to need to verify as we move forward?
Yeah, totally. It's an important question. And so imagining that I had, performed that experiment or seen these results without anybody ever having studied, circadian gene regulation, then the question is, what do we believe? So the motifs that I put up there, it's also worth noting, are, half a dozen out of several hundred. I do think it's possible that there are diamonds in the rough among the 700. I'm going to sort of punt on that question. We do it through collaboration with people who know more than I do about molecular biology and how to chase down these sorts of leads, because that's really not my domain of expertise.
Thank you for the talk. It was very informative. So when you're looking at how location impacts like phenotype and how gene regulation is working in these crops, how do you factor in the microbiome? Specifically, say there's like disease pressure further south that we don't see in Wisconsin.
Totally, yeah. So right now, we are not explicitly modeling microbiome effects at all. So when we talk about environment, we are just integrating over everything within a particular location, whether that be microbiome, weather, soil type, who planted it, and how they planted it. Moving forward, though, we do have some work going on seeing if we can kind of start to parse microbiome out from abiotic stresses and host genotype. I don't have any good answers yet, but they're hopefully forthcoming. These are projects we're just getting off the ground right now. Be happy to chat more about it.
Really good talk. So you emphasize a lot looking at G/E across multi-environment testing, and then you also, I think, really focus on the temporality of how these phenotypes and gene expressions change across time. Have you given any thought, or do you think there's importance in kind of that sort of microtemporal scale of G/E, where how phenotypes change or gene expression changes, like even between days, within the growing season, if there's a stress event that happens, like even on a 2 to 3 day scale, how the dynamics of that?
Totally. Yeah, I think that's a really interesting sort of topic to dig into. It's hard because sort of the finer your granularity, kind of like the lower your power goes or the more your experimental units get spread out. We did an experiment this past year where we put temperature sensors in individual plots throughout the growing season and have some kind of like interesting preliminary data about that or from that shows that it's quite possible that morphological differences between inbred varieties are sort of, I won't say affecting, but correlated to differences in temperature regimes throughout the season. We haven't done any digging into that data set to look at sort of like spatially confined phenomena or like the temporally resolved outcome of like extreme events, but the data are there to start playing with. something that I'm learning as a relatively new PI is that it's easy to generate data and it's hard to analyze it. So like we're sitting on tons of stuff like this and I just don't have the people to do it. Yeah.
Great talk, Joe. Thanks so much. So obviously you're a big corn guy, and that's awesome. But a lot of these same problems are being faced in like different crops at the same time. Do you see any way for machine learning / AI to kind of help bridge that gap between species, and how can we use information from one to predict the other?
Yeah, 100%. And I like, are you an audience plant? Because that's actually something that I meant to address in the talk, and I just forgot to say it. So thanks. Where to start? So the latent phenotyping models, I think, are particularly useful because they have the potential to be applied to whatever you shove into them, which, garbage in, garbage out. But if you can collect high-dimensional data on your system of choice, you may have sort of a pre-baked way to generate traits that may or may not be of interest to you. And so part of that project actually that I forgot to mention is we are really focused as an end product on developing tools for the community where people don't need to have domain expertise on how to build and tune these models in order to use them. So that's kind of on the phenotyping side. You specifically asked about gene regulatory side. I think the fact that we see this convergence between maize results and Arabidopsis, previous findings in Arabidopsis, is a really promising indicator. Those two species are separated by something on the order of 200 million years. And so, there are aspects of gene regulation that are really inherently conserved, if not how they perform on specific genes, then the way that they act in general within the genome in order to affect change. And so learning about them in one system, I think, gives us power, hopefully, to apply them to other systems. And that's something that I am very interested in pursuing, but not actively doing quite yet. I think we're still sort of building the data sets that we need to train the models in order to start porting things into other systems.
Joe, thanks for over here.
Yeah.
I'm not a geneticist, so let's go back to phenotyping. You talked a little bit about leaf orientation with corn, and I have a series of questions on this. We're at a Corteva Symposium. They've recently talked about orienting seeds in the furrow to mechanically change the orientation. So is the genetic difference in leaf orientation more powerful than us physically manipulating the seed? And what's the value of doing that?
Yeah, that's a really insightful question. So the sort of original study that I pointed to that is published, done by Jan at Iowa State, I know he was very careful with thinking about how orientation of the seed impacted the phenotype that they were measuring, and they spent quite a while sort of parsing that out in controlled environment conditions before going into the field. I don't know if you had to proportion variability for the phenotype to planting technique versus genetic influence. I don't know. Maybe we can talk to some of the Corteva folks about it, but beats me.
Quick follow-up. How do you think the introduction of short-statured corn hybrids is going to change the way you do phenotyping? And have you interacted with that at all? And is that going to change the way you think about phenotyping?
I haven't worked with them at all. Just off the top of my head, having sort of a shorter canopy space might be sort of a technical advantage for phenotyping. One issue with using UAVs is sort of penetration through the canopy. Although I actually don't know if short corn has denser canopies, in which case maybe it's moot. But that's at least one way I can think of that it might be kind of nice that we can sort of see farther down. Yeah. off the hook?
We got one more question at
Okay.
least, because we got a little bit of extra time before we move to the next speaker. So
should
I
have talked
was
more.
I was going to ask you on the slide where you were showing the partitioning of the variance on the transcript abundance.
Mhm.
Have you looked at separating that in core and accessory genes? And have you looked at that? And if you haven't, do you think you would see a pattern in how those variances are partitioned in both of those groups?
We haven't split them out yet, but that should be pretty easy to do. I think it depends a little bit on your interpretation of what's in those core and non-core groups. I sort of have a hunch that Ty may have thrown away a lot of the non-core genes already based on sort of low levels of expression. I think those are kind of enriched for ongoing pseudogenization and things like that. But I, off the top of my head, quick guess. I think some of those core genes have been guessed to be involved in, tolerances to particular like biotic or abiotic stresses. And so we may see that they show sort of genotype-specific spikes in expression in particular environments where they're relevant. Be a fun thing to look at, and I think we could figure out pretty quickly by plotting out some sort of like positive controls, sort of how they're acting. Be fun.
Awesome. Thank you very much, Joe. With that, I think Audrey. Yep. Okay. We're giving you the green light to call it good. Thank you very much,
Thank
Joe.
you so much.
And if we did not get to your questions that you had submitted on Slido, we will get to those in the Q&A panel. So don't worry about that. Thank you for submitting those. With that, let's move on to, it's our break. There we go. So we will take a short break and we're going to reconvene back in here at 11 o'clock. So thank you. Enjoy the refreshments outside.